morning. Thank you to the OIS for having me today and introducing Eyevensys technology. Last time I presented was before the Academy. Eyevensys is a French company incorporated in Paris, but also incorporated here in the United States as of early this year. The founder was Dr. Francine Béard-Cohen in Paris. We are in the middle of raising our Series B, 30 million euros. We already secured 22.5 last month. The question we're addressing is really try to answer the delivery of protein in the back of the eye, try to avoid subretinal injection, try to avoid intravitreal injection, try to avoid systemic administration or ocular implant. So the technology consists of plasmid, which are encoded to produce therapeutic proteins. These plasmids are injected using a little ocular device in the ciliary muscles and through the system of electrotransfection, which you probably are very familiar. The idea is to allow this plasmid to integrate with the ciliary muscle cells and be able to produce therapeutic proteins directly in the vitreous, avoiding repeating injection and having a sustained delivery. So to validate the technology, multiple animal models have been tested, of course, with different proteins. And in preclinical models, we were able to show that we can encode plasmid to produce any type of proteins. We were able to show durability. That means producing this protein for a long period of time in several animal models, in rats, rabbits. We were able to produce anti-VEGF protein, neuroprotection. And the company decided to go with an anti-TNF approach to validate the platform. It was tested in animal models first, successfully. This is the first indication we are actually in the clinic. I'm pleased to report today that we are in a clinical trial in France and in the UK. This is a dose-escalating studies. These are patients who have failed multiple treatment, especially steroids, for multiple years. Vestroia G, the goal here was really to demonstrate safety of the technology, but we were able to put this device on the eye to do electrotransfection without damaging the eye and able to produce protein. But we did not expect to see efficacy. However, we are very pleased to see that the last two patients, the last cohort, the last highest cohort, they were able to show one patient at 6 months, so one injection at 6 months. We show a 12-letter improvement for one patient on the left, and also a decrease in edema. The patient on the right also has a reduction in edema. I didn't have a slide ready for this presentation, but I got the data yesterday for the 8 months visit. And at 8 months, we see a 14 letters improvement as well as a totally nice flat OCT. So what are we saying with that, with this statement? What we're saying is that we are able to produce protein in the vitreous, obviously, by injecting these plasmids. We are able to have sustained delivery because we did one injection, and after 8 months, we're still able to produce protein, and we're able to see efficacy in patients who had failed since several years already treatment with steroids and anti-TNF alphas. So we are cautiously optimistic. The next indication we are tackling and looking into is retinitis pigmentosa. The approach here is to look at trapping the iron, which is produced when the photoreceptors are dying, using human transferrin. That's a protein which is natural in the eye. And we have several preclinical models which show that we are able to produce a protein, protect the outer segment of the photoreceptor, and have sustained delivery. For this project, we are launching our IND-enabling phase right now. The device, of course, we have two devices that makes it a little bit more difficult. One is an ocular device, one is a generator. We are launching the uveitis studies in the US. We're filing an IND probably next week. We just finished all the testing. And we did finish all the testing for both devices to be compliant with FDA guidance. So our pipeline, again, uveitis in clinical trial, ready to launch the clinical trial in the US here. RP, launching the IND-enabling phase. We're also looking at AMD, of course, with anti-VEGF. And the team is an experienced team coming from pharma. Many of them, you know, I joined Eyevensys 2 years ago. And on the board are our investor and some of the faces that I'm sure you will recognize. That's it. Thank you very much for your attention.